Hi, everybody. Thank you for Tico for your invitation to speak at this conference. So today I'm going to talk about data science for sustainable agriculture at Bayer Crop Science. And specifically, I like to introduce you guys a Bayesian modeling use case and how we can couple that with visualization. So a quick round of introduction. So I like to just share with you a little bit about my role. So I work cross-functionally with a team of data scientists and business partners to build basically digital agriculture solutions. So my role where I come in is as a technical lead, I try to coordinate and also develop different technical solutions using predictive models and optimizations to create tailored solutions for our business partners. I'm also a founding member of Bayer's Women in Data Science organization, which I'll talk a little bit about later. Our goal there is to make data science a more collaborative and inclusive space. My journey, I am from Macau, and you can see here, this is on the top, this is where Macau looks like. And then I moved to Wisconsin to study in college and also in grad school. And finally, as of 2015, I've moved to St. Louis, where I have also completed an MBA and start my job here at Bayer Crop Science for the last 5 years. Outside of work, I am a musician. I like to play classical piano. I love to cook, and I also like to hang out with my cats. And so you can see a picture of my two cats, and here's me trying to play the piano and get some tips from the mall. But that was actually a friend that was staging there. So, well, enough about me. I like to actually talk a little bit more about why I'm here and why what message we like to really send through this talk. So one of the important thing I've learned as a data scientist working in an agricultural company is how many decisions there are when it involves, planting and growing something and creating these organic products, right? So, well, traditionally, agriculture isn't widely thought of as an industry that is associated with large scale data, analytics, or artificial intelligence. But I can tell you from my own ignorance growing up in a largely urban area, it wasn't really something I ever thought about. I mean, I love food and I like to try my hands at gardening, but growing tomatoes in a little pot is one thing, even though I'm not very good at it. But how do we grow enough food to feed our world's growing population? Well, this is an enormous network of problems that people are solving actively every day. But starting with a farmer who is growing corn on their family farm, this means making 40 to 50 decisions every season, and you have to make them correctly to provide the best chance of having a successful crop at the end. As you can imagine, this is no easy task. So in the past, many of these decisions are done based on experience, tribal knowledge, and just sometimes luck even, right? But one thing we know for sure is that farmers are extremely resourceful and innovative. So of course they come up with something to tackle this problem. So here on the left, you can see it is called a flamethrower. So that's one of the solution to our problem of growing food on a large scale. So this flamethrower on the left, what it does is it helps to stunt the growth of plants. And on the right is the detasseler, which artificially turn a corn plant into a female parent for hybrid corn production. So basically, farmers get to work with dragons and transformers for their job, which is super cool. This, however, comes with a heavy responsibility. That is, in just 30 years, we are expected to grow 70% more food to feed the growing demand of our population. So imagine how challenging it is already to feed the world as it is right now. And when we factor in the limiting resources we have on Earth, it almost seems like the only solution is to start growing plants on Mars. Well, until we figure out how to do that, another approach is to tackle this challenge by utilizing data to help us make better agronomic decisions. So this is where our data science community comes in. And I also like to tell you a little bit about the data science community at Bayer Crop Science. So we are a growing community of data scientists from all walks of life around the globe, and we have a common goal to feed the world in an innovative and environmentally sustainable way. Um, Before the pandemic, we really big on events and training workshops and hackathons, and we really want to make sure we are, good partners to our business, to our business partners, to our growers. But at the same time, we want to have fun and solve, real problems. And this is our way to come together by being present at different conferences. But we don't stop there. A big part of being a true data science community is making it more inclusive, collaborative, and also more equitable. And so from there, we have one of the big challenge, right? So data science is a new field that emerged primarily from computer science, engineering, and math, which for one reason or another, it means that there has been less female representation in the past. And this really shows. So a survey done by Boston Consulting Group found that as of 2020, there are only 15% of women in the data science field. At the same time, we're building tools that are going to affect people's lives. And that means people from all gender, ability, ethnicity, and cultural background. So it only makes sense to have people build tools with people that are representative of our own community. And so at Bayer, we have started this initiative called Women in Data Science with a core set of missions to inspire, attract, grow, and champion women and our allies who are pursuing a career in data science. We have hosted chapter conferences in collaboration with the Stanford WITS program and also internal awards programs with the aim to recognize women data scientists that are doing outstanding work and support one another. So to that end, I like to share with you a project I've worked on as a data scientist, which is a use case about seed treatment trials. First, a little bit background of corn. So if you're from the Midwest of the US, you might have heard a saying called knee high by the 4th of July. This is referring to the massive acres of corn that we grew in our heartland, which is also what we call our corn belt. But underneath those plants, maize for Halloween, it is interesting to also think about how these things grow. So, well, there are two parts, okay? So above ground, obviously, we have our weather condition that really affects how, the health of our fields. But what is also equally crucial is the environment, the underground things that these plants grow in. So our soil and the organisms that comes with it plays an equally crucial part to how tall or how healthy these corn will grow through our entire season. So some of these underground organisms is called the microbiomes, right? So the microbial community is super important, and this is what we'll pay our attention to for this talk. So zooming in to look at this a little bit more, we are interested in understanding how a plant communicates with these organisms in the soil. So we call this our microbial community, as I mentioned, and there are two ways, right? So there is the rhizosphere, which is a layer of soil that is our communication channel between a plant and the soil microorganisms. This sends signals to the plant tissues that will then influence its phenotype, such as yield and disease resistance. And if we can understand how communications work between the soil and the plant and how it interacts with the environment, we will be also be able to create treatments that can help us influence these different phenotypes to help the plant thrive. And here are the questions. How do we pick the treatments? What kind of corn variety and which environment work best together? So to answer that question, it is really important to start with data. So the question is, how do we get these data? Well, we start with an experimental design. And here is an overview of our process. We start with scouting locations, designing the sets, applying treatments, growing these plants. And the fun part, of course, is when we get to play with the data. Now, another question is, how do we do that? Well, we start with an assumption based on what we understood about the process. So in this case, the coin growing process, and what kind of factors or covariates contribute to the variation in the observations we see. So with the process assumption and the model error, this is represented mathematically by a model that looks like this. So you can see, for example, Z is our observations, Y is the function of the factors, and epsilon is the random error. So with that general framework in mind, the first step to do as a data scientist is to start with something relatively simple. So the first step we tried is to build a linear mixed model. And so what that means is we are considering the observations being influenced by the different microbial treatments and environment separately. So you can see these different effects will form kind of parallel lines in the regression model. And what does that, what does that tell us? So the next question is, how do we diagnose these model impacts, right? Based on our initial assumption. Well, so the first result we can see, so it's a pretty busy slide here, but on the top left corner, we see the actual yield of a plot. And below that is the predicted yield. So if we look at just, from these two pictures, you may say, okay, well, the prediction is not too bad. So the model is doing a decent job capturing the yield, right? But if we switch over to the column in the middle, the model residuals are showing a very interesting spatial pattern. And now when we look at the model residuals and comparing that with an identity line, which is on the middle, the bottom middle figure, you can see that the model residuals are not really, are kind of biased from the identity. And also moving over to the third column, you can see that there is a highly skewed distribution for the model residuals. And what that means is that the model error are not independent in this case. And so it is telling us that maybe there's something missing in the model development process. So we're not really capturing all the variation in the model itself. And therefore, some of these variations are actually showing up in the model residuals. So what next? Well, we have to think about, why that would be. And one of the one of the obvious thing that probably jumps out immediately is the nature of this trials. So because we are designing experimental trials, and by nature, these are spatial data. And so we actually have to, also account for spatial variation in the model building process. So in the first model, we've really only looked at the treatment type and the environment as if they are separate and independent, but really they should be interacting with each other. And then within each environment, we should take into account how each plot should relate to one another in terms of their variation in the observed yield. So here is a second attempt, which is slightly more complex, but the difference is in addition to the two fixed effects, which is the x1 beta 1 and x2 beta 2, we've added two more terms, which is a model impact that looks at how the variation within field and across field should be contributing to our model observations. And what is special here, just to point out, one of the coefficients is actually showing us how how the variation between plots should be impacted based on the distances we see. So on the figure in the lower left corner, we can see that the spatial correlation between these observations is very close when you have two plots that are close to one another, and it's start to go down as the distance increase. So that relationship is also captured in how we model the effect, right? So the effect is stronger when these two plots are close together and weaker when they're further apart. So that is the spatial component of our Bayesian hierarchical model. And now the second part is a Bayesian hierarchical part, right? And what that means is in addition to adding this spatial component to address the spatial correlation as a function of distance, we also want to look at how we define the model effect for the treatment and the interaction. So in here, what is highlighted in orange, these are the coefficients for the effect. So basically, we will tell you, okay, if I think the treatment deserves a higher effect, maybe I'll give it a number, right? Let's say it's 3 parts in a treatment and 2 parts treatment by environment. Well, another way to think about this is we can think of this effect as a distribution. So maybe beta can be a normal distribution that depends on a variance. Now, this variance parameter is also depending on another distribution. So you have a sort of a Russian doll effect on how we can specify the requirement of the model based on our understanding of the process, and also some assumption based on some expert knowledge. And with that, what that looks like in the model is actually making quite a bit of difference. So here's the results. Now, when we have our Bayesian hierarchical model, we address 2 things. One is we address the spatial dependencies between each plots. The other thing is we also address the complexity of the parameters in our models. So what that means is the modeling effect attributing to the treatment or the environment or the interaction between treatment and environment is not just a fixed number. It is actually a nested system of quantities that we can fine tune based on the different distributions we choose. So that is the Bayesian part and the hierarchical part. And now you can see as a result, right, we're looking at the same plot of yield data. And if we take a look at the error in the middle, so you can see that the model residuals are much more randomly distributed. And this is exactly what we want. So we really don't want to see patterns in the model error, because error by nature should be random. And this is what we're seeing, right? So that means, this is going to work. This is actually more or less capturing the observations accurately. But there's only one last issue is we have thousands of plots for thousands of products that we analyze as part of these C treatments trials. So how do we visualize this quickly, efficiently, and help us make processes more easily? Because I can't really go through thousands of slides like this, and this is where a visualization tool like Spotfire comes in. Now, so putting this all together, we have really 3 things we can do. So we can start with how to connect our data with the data wrangling function through the register data functions. So basically, we can build in R scripts, basically running the model and connecting the data from an S3 bucket. And we can also generate different custom columns depending on what we're looking for in the report. And we can generate these analysis pretty easily based on the recommended visualizations and customize it based on what our business partners are using or are looking for. And based on that, our business partners who are researchers in the R&D pipeline can make informed decisions to decide, which treatments are showing the effects that they would like to see. And this is how we can maybe advance the next round of experimental studies. And so here is a demo for our dashboard for this C treatment trials. So in this video, you can see if I go to the map view, I can pick a few treatment locations and see what is the model effect from that particular treatment to in a particular location. And we can also see if we go to a different window, and if I just pick a specific plot, and I can see in these plots what are the different environmental factors, also what the raw data of the plot looks like. So this really helps me to visualize sort of the interactive effect of the model observations, the model effect, and also what the treatment interactions look like. So this is really helpful for a user like a researchers in the R&D development pipeline to look closer and also get the kind of information about, the covariates that we're using to build the model to help them answer questions like, if I have these products grown in an environment with a very low pH level, which is the top right corner, what does that mean to my model effect? And if I have a plot in this location in the Midwest, for example, what does the treatment effect generally look like? So that is really, really helpful for us to have this spot fire. And as a takeaway, I just like to say there are a couple of things that are really important for this project. So, and this may be common to many other data science projects as well. First is we got to experiment, and sometimes we fail, sometimes we win. But the first step is really to try something and learn from it and repeat the process. The second piece is through that experimental analysis, we can get to know our data. We really can understand our data by how we model it, by understanding the error. And with, by paying attention to model error, this also helps us better, communicate our model, and of course, communicating our results through visualizations so that the stakeholders or our business partners or research collaborators can understand the story that we're trying to tell. And with that, I'd like to say thank you again for the conference organizers, and I hope this helps. Thank you.